Welcome students of the Maryland University for Integrative Health. This is Dr. Bill Gurley at the University of Arkansas for Medical Sciences, College of Pharmacy. And my talk today is entitled Multi-Ingredient Caffeine-Containing Dietary Supplements, Their History, Safety, and Efficacy. Now my purpose today is hopefully to instill in you a better appreciation for the pharmacological consequences that can emerge when natural caffeine sources are combined with a host of other phytochemicals. Over the years, I have often referred to such multi-ingredient caffeine-containing combination products as veritable pharmacological Pandora's boxes. Now, all of you in the listening audience are well aware of natural caffeine sources like tea and coffee, as well as their role in both Western and Eastern society. Natural caffeine sources in their own right not only taste good when properly prepared, but are, for the most part, fairly innocuous and even have some positive health benefits. Over the last 2 decades, however, several new classes of caffeine-containing products have burst upon the commercial stage, and their effects have taken the medical community by surprise. While probably not historically accurate in every regard, this timeline puts into perspective the recent appearance of multi-ingredient caffeine-containing dietary supplements onto the world stage. Now, to understand these products, one must have an appreciation for the pharmacology of caffeine. And caffeine is one of the most widely consumed and heavily studied stimulants in history. As a phytochemical, caffeine is unique because it has an excellent oral bioavailability, it has a relatively small apparent volume of distribution, and is not highly protein bound. Therefore, it readily crosses the blood-brain barrier. In the liver, it's metabolized primarily by cytochrome P451A2, and its principal metabolite, which is known as paraxanthine, is also active. The dose-dependent central nervous system and cardiovascular stimulant effects are primarily due to the fact that caffeine is a non-selective adenosine antagonist. It's an antagonist at both adenosine subtype 1 and adenosine subtype 2A receptors. Now, as concentrations of caffeine in the blood increase, it can also become a non-selective inhibitor of various phosphodiesterases, which lead to the accumulation of cyclic AMP within certain cells. At even higher concentrations, caffeine can activate ryanodine receptor channels and stimulate the release of intracellular calcium. And also at these relatively high concentrations, it can also inhibit GABA-aminobutyric acid neurotransmission. And finally, as many of you are probably well aware, tolerance to caffeine's pharmacological effects can develop with chronic exposure. Now, as a general rule, caffeine-containing beverage have an excellent safety profile. The safety concerns with caffeine-containing beverages are typically associated either with overindulgence or to individual sensitivities to caffeine. The safety profile for multi-ingredient caffeine-containing dietary supplements, however, is much less clear. A search of PubMed and Web of Science indicates that since the early 1980s, there has been a steady increase in publications related to adverse effects related to caffeine. Since 2000, the number of publications related to adverse caffeine effects has more than doubled. This increase is undoubtedly linked to the upsurge in novel caffeine-containing dietary supplements as well as energy drinks. So this data certainly indicates that we are truly a caffeinated society. Now, there are a host of reasons why multi-ingredient caffeine-containing dietary supplements have been linked to adverse health effects. As I mentioned earlier, oftentimes overindulgence or just an individual that may have an underlying health condition may make them more sensitive. Many times these dietary supplements are taken concomitantly with other prescription stimulants and pressor agents like the amphetamines or methylphenidate. Oftentimes they are formulated with other naturally occurring stimulants or other naturally occurring pressor agents, for example the ephedrine alkaloids or synephrine or other phenylethylamines and even the compound yohimbine. Sometimes these dietary supplements are adulterated with prescription or illicit stimulants and pressors. Classic examples are sibutramine, as well as dimethylamylamine and beta-methylphenylethylamine. And of course, consumers expressing genetic polymorphisms of various receptors and enzymes and transporters that are involved in caffeine disposition may also be overly sensitive to the effects of caffeine. And so it's not uncommon for individuals that may have polymorphisms for the adenosine receptors or for catecholomethyltransferase, which is involved in the breakdown of norepinephrine, which oftentimes is stimulated by the release of caffeine. There also can be mutations in the drug metabolizing enzymes and a wide variety of other proteins and receptors that caffeine may affect. And of course, it's not uncommon to see adverse effects associated with these types of products when they're taken in combination with vigorous exercise. And what's really ironic is that many of these products are actually marketed to be taken in conjunction with vigorous exercise. Now, I want to briefly describe a few of the issues associated with the safety and efficacy of these products using examples from these three general categories. The ephedra-containing dietary supplements, which are no longer on the market, the replacements, which are the ephedra-free dietary supplements, and also we'll talk about energy drinks to some extent. Now, the ephedra-containing dietary supplements provide a good opportunity to illustrate the concept of caffeine-mediated pharmacodynamic herb-herb or herb-drug interactions. And from 1999 to 2005, the medical literature was awash with case reports describing cases of heart attack, stroke, seizures, and psychotic events that were linked to ephedra supplement use. And three of the more popular ones that were involved in associated with a lot of these adverse event reports were Ripfuel, Metabolife, and Xenadrine RFA, as pictured here in the lower right-hand corner. Now, one of the primary reasons for the problems associated with ephedra was the combination of ephedrine alkaloids and caffeine. Now, synthetic combinations of ephedrine alkaloids and caffeine, which were known as amphetamine lookalikes, had been banned by the FDA back in 1983. And because natural combinations of ephedrine alkaloids and caffeine fell under the definition of a dietary supplement, they remained on the market for over a decade, from 1994 until just after 2004, until the FDA could prove that they were harmful. Now, ephedra species are natural sources of ephedrine alkaloids, like ephedrine, pseudoephedrine, methylephedrine, norepinephrine, and norpseudoephedrine. Norpseudoephedrine just happens to be a Schedule IV stimulant. Like most botanical supplements, the content of the phytochemicals can vary considerably as a result of several factors. Ephedra-containing dietary supplements were often formulated with a multitude of other botanicals, many of which were natural sources of other stimulants. And some examples included Coleus forscolii, Citrus aurantium, and yohimbine. Now, here are the structures of several sympathomimetic amines. And you can see that the second structure is ephedrine, and it's very similar to methamphetamine at the top. Actually, sometimes ephedrine is referred to as hydroxymethamphetamine. And then synephrine at the very bottom is one of the latest ephedrine substitutes. So you can see just from a structural perspective how many of these sympathomimetic amines are quite similar. And so from a structural perspective, but also they're similar in many regards with regards to their pharmacodynamic effects. Now, during what I like to call the ephedra wars, manufacturers often touted the safe track record of ephedra use in China. Unfortunately, the Chinese used ephedra very differently than how it was used in the United States. So the Chinese had been familiar with ephedra for over 5,000 years, and typically they used it to treat asthma and nasal congestion, but they rarely indicated that it be used for more than 7 to 10 days. They almost never combined it with other stimulants like caffeine. And it was typically administered as a decoction, or they would brew it up as a tea to be consumed slowly. And they never indicated it to be used as a diet aid. Now compare that to how the ephedra-containing dietary supplements were used in the United States. Again, they had an extensive use from 1994 to 2004. They were marketed almost exclusively as diet aids or energy boosters or exercise performance enhancers. They were indicated for chronic use, and they were almost always combined with other stimulants, primarily caffeine, and they were administered as concentrated extracts in the form of tablets, capsules, and concentrated liquids. So the Chinese learned quite a bit about ephedra over that 5,000-year period. We, as a society, just failed to heed any of their advice. Now, ephedrine and caffeine potentiate each other's cardiovascular and central nervous system stimulant effects. In fact, the combination of ephedrine and caffeine has about 1/5 to 1/10 the potency of amphetamine. And there are a number of phytochemicals that are present in many of these botanical supplements that can potentiate the activity of these of the ephedrine caffeine combinations themselves. And so phytochemicals like the catechins and xanthones and certain tannins that are present in natural caffeine sources, again, can potentiate the sympathomimetic activity of both ephedrine and caffeine by inhibiting some enzymes, primarily catechol-methyltransferase or monoamine oxidase. Moreover, ephedrine and caffeine is one of the few drug combinations that have been known to significantly lower cerebral blood flow. And it's this combination, or this pharmacologic effect, that may underlie many of the ischemic strokes that have been associated with the use of these particular products. Now, it appears the complexity of ephedrine-containing dietary supplement formulations was the source of many of the problems. So I'd like to illustrate how the more complex the formula was, the more toxic it could become. So here are 4 ephedra supplement formulations and their ingredients. Now, apart from EPH833, which only contained ephedra, all of the others incorporated guarana as a natural source of caffeine, as well as a host of other phytochemicals that could potentially interact with the ephedrine alkaloids. And here you can see the complexity of these particular products, Metabolife, Ripfuel, and Xenadrine, all of which had guarana as a natural caffeine source. Now, what I want to do is utilize this table, which is part of a toxicity pilot study with some of these ephedra-containing dietary supplements that was conducted in mice. And in this phase of the study, there were 4 mice in each group, and each mouse received 400 microliters of an aqueous extract of each one of the products via oral gavage. Now notice that the EPH833, which I've highlighted in orange, which only contained ephedra extract, produced no mortality, even though the ephedrine alkaloid dose was 3 to 5 times greater than that of the other products. And if you'll notice that the more complex formulations, which had lower ephedrine alkaloid content, produced much greater mortality. 3 of the 4 animals that were in the Metabolife and Ripfuel group succumbed within 72 hours, whereas all the animals within the Xenadrine group succumbed within 24 hours. So clearly, for the products with more complex formulations, there was significant mortality was achieved with much lower doses of the ephedrine alkaloids. So again, the point is that it wasn't just the ephedrine alkaloids, it was the whole milieu involved in these particular products. Now, as a follow-up to the mouse pilot study, we also performed a pilot study in Sprague Dawley rats. And the rats were surgically implanted with radiobiotelemetry devices that allowed us to continuously monitor cardiovascular parameters like blood pressure, heart rate, and EKG activity. Now, 3 days after the surgery, the animals were administered 1 milliliter of an aqueous extraction of either 1 EPH833 capsule or 1 Yellow Jacket capsule via oral gavage. And the cardiovascular parameters were monitored for 24 hours. And after that time, the rats were euthanized and their hearts and brains and other tissues were harvested and examined histologically. Now, here are the doses of the ephedrine alkaloids from the 2 preparations, as well as the composition of the formulations. The EPH833 again only contained ephedra extract, whereas Yellow Jacket contained kola nut, capsicum, ginseng, and citrus aurantium. Notice that the ephedrine alkaloid dose for the EPH833 was almost 4 times greater than that of the Yellow Jacket. Now, these are just baseline EKG tracings from representative animals in each group prior to dosing them with either the EPH833 or the Yellow Jacket. And you can see that the EKGs are fairly similar. Now, these are EKG tracings from representative animals in each group 1 hour after dosing with each preparation. And the EKG tracings for the EPH833 are no different from baseline, whereas those in the Yellow Jacket group are starting to show signs of premature ventricular contractions. At 4 hours, the EKGs from the EPH833 group again remained unaffected, whereas the yellow jacket animals are showing signs of significant cardiovascular distress with marked electrophysiological changes. At 5 hours, the EKGs from the EPH833 group show no untoward effects, whereas the yellow jacket animals are exhibiting what we refer to as the death rhythm, which was an EKG tracing that baffled the cardiologist. When this rhythm developed, it would invariably lead to death, and we would euthanize the animals at this point. And histopathological examinations of both the hearts and brains from these animals showed evidence of myocardial infarction as well as cerebral ischemia. Now, one of the reasons you start to see a lot of these effects is that the two principal pharmacologic agents in those products, caffeine and ephedrine, reach their peak concentrations within the blood at about the same time. So this figure on the left represents plasma caffeine concentrations at the top and plasma ephedrine concentrations at the bottom after healthy volunteers were administered an ephedrine-containing dietary supplement product that containing both caffeine and ephedrine. And you can see in the pink shaded areas how the peak concentrations occur at about the same time. And so again, the ephedrine alkaloids and caffeine from ephedrine dietary supplements are not only very bioavailable, but they exhibit very similar Tmax values, and Tmax is the time that it takes to reach the maximum concentration in the bloodstream. What was really interesting is that the toxicity for a lot of these case reports were exacerbated in conjunction with exercise. And it just so happened that exercise and the ephedrine-containing dietary supplements were a common feature of many of the adverse event reports and case reports that appeared in the medical literature or that were submitted to the FDA. Now, with regard to efficacy, the ephedrine-containing products did produce mild to modest weight loss in some individuals, but oftentimes only when combined with a low-calorie diet. Untoward, but not necessarily serious side effects like elevated blood pressure, tachycardia, insomnia, and such were frequently encountered. Nevertheless, certain consumers appear to be more susceptible to serious adverse effects of ephedrine-containing dietary supplements, again, especially when combined with vigorous exercise. Now, at the peak of the ephedra controversy, there were several highly publicized deaths of college and professional athletes, which dramatically affected the sales of ephedra-containing dietary supplements. And then after their peak around 2002, sales of ephedra-containing dietary supplements started to plummet. And the manufacturers of ephedra-containing dietary supplements simply just took the ephedra out and started marketing ephedra-free dietary supplements. And pretty soon, those ephedra-free supplements caught up with and surpassed their ephedra-containing predecessors with regards to sales. And so after the FDA removed ephedra-containing supplements from the market in 2004, ephedra-free supplements quickly took their place. And since that time, eerily similar case reports and adverse event reports began appearing in the medical literature. And many of these case reports were documenting almost identical effects that had previously been associated with ephedra-containing dietary supplements. So perhaps ephedra wasn't the whole problem. Now, we recently conducted a small clinical trial with 4 ephedra-free products in 4 healthy volunteers, and the 4 products were Xenadrine EFX, Xantrax 3, and Metabolift, and all of them contained guarana as one of their natural sources of caffeine. So we utilized a guarana supplement somewhat as a comparator. You can see the complexity of the various dietary supplement formulations, and highlighted in pink there are the dose of caffeine per capsule and the cumulative dose that would be received in a 24-hour period if the label directions were followed. And so in our study, we utilized 12 healthy male volunteers. They were randomized to receive each one of these 4 products over a 6-month period of time. And during the active phases of the study, each individual received 2 capsules of each product 3 times a day for 3 days. And we monitored each subject for blood pressure, heart rate, and EKG activity. And we used Holter monitors, which are just portable EKG systems that you can wear under your shirt to collect EKG data over a 24-hour period. And what we saw were significant increases in systolic and diastolic blood pressure as well as heart rate. And during the active phases of the study, we saw much more abnormal EKG activity as opposed to during the washout phases. There was also a lot of complaints from the subjects regarding nervousness, insomnia, as well as nausea and vomiting. And so clearly the caffeine dose and the complexity of the formulation likely contributed to the untoward effects that we observed in our study. What was interesting is when we were analyzing the products for caffeine content, two of the products literally ran us out of the lab because they smelled so bad. And my technician commented that, we may have a bacterial contamination problem. So we sent the samples off to the National Center for Toxicological Research, and sure enough, two of the products were heavily contaminated with bacillus species, one of which was Bacillus cereus, which is a human pathogen. Now recently, another dietary supplement known as JAK3D, containing a sympathomimetic amine known as dimethylamylamine and caffeine, has been linked to numerous case reports and adverse event reports, including deaths of several active duty soldiers. Now, dimethylamylamine was developed by the drug manufacturer Eli Lilly in the 1940s as a nasal decongestant, but it was removed from the market in 1983. Now, JAK3D combined DMAA with caffeine and several other botanical extracts and was marketed to be used in conjunction with vigorous exercise. Again, sympathomimetic amines, caffeine, and exercise can be a bad combination for some consumers. Now, most consumers don't have a clue as to the function of many of the botanical extracts found in dietary supplements, but ingredients certainly have consequences. Now, as I mentioned earlier, DMAA is a synthetic sympathomimetic amine that's metabolized by an enzyme called CYP2D6, and one of the components of JAK3D was a botanical extract called Schizandra chinensis. But why would you want to add Schizandra to the formulation? Well, Schizandra just happens to be a botanical that contains a number of these methylene dioxyphenyl containing phytochemicals that we've mentioned in a couple of our other lectures. And some of these methylene dioxyphenyl containing phytochemicals can inhibit various cytochrome P450 enzymes, including CYP2D6. And there's some evidence to suggest that DMAA is metabolized to some extent by CYP2D6. And so by inhibiting DMAA metabolism, you may increase its oral bioavailability and therefore also increase its elimination half-life, producing more accumulation of the drug and potential toxicity. And so again, by increasing the oral bioavailability plus adding caffeine, you can increase the toxicity of DMAA. And this is just one of the phytochemicals found in Schizandra, one called gomacin, but there are a whole host of others that look very similar to this, but again, they have those methylene dioxyphenyl moieties, and it's those types of phytochemicals that are notorious for inhibiting human drug metabolizing enzymes, particularly the cytochrome P450 enzymes. And so it's suspected that these methylene dioxyphenyl containing compounds may have altered the metabolism of DMAA and contributed to the exacerbated pharmacodynamic effects of DMAA and caffeine. Now, DMAA was once thought to be present in geraniums and was actually called geranamine, but it's been found out recently that this compound does not naturally occur in geraniums. But nevertheless, in this figure, the combination of caffeine and geranamine, which is also known as DMAA, has a more profound effect on systolic blood pressure than just caffeine alone. So caffeine alone is shown at the bottom with the green circles, and then caffeine plus DMAA at the top, looking at the effects on change in systolic blood pressure. And you can again see that the combination of caffeine and geranamine is much more potent than just caffeine by itself. So again, these complex formulations can oftentimes have complex effects. Now, with regard to efficacy for some of these ephedra-free dietary supplements, there have been many more safety and efficacy studies conducted on ephedra-free dietary supplements. The results of these studies are often disappointing with regards to efficacy. Their safety profile is still up for debate because most of the studies have only investigated either a single dose or utilized doses that were well below that recommended on the label. Given the frequency and severity of the extant case reports, Just because a product is ephedra-free may not necessarily mean that it's trouble-free. Now, in many respects, energy drinks are a lot like ephedra-free supplements, and not too surprising, case reports and adverse event reports associated with energy drinks are eerily similar to those linked to ephedra and ephedra-free supplements. And so caffeine toxicity and interactions of caffeine with other components likely underlie many of these reported adverse events. Now, it just so happens that energy drinks are one of the fastest growing supplement categories. They rank only second to vitamins in popularity among adolescent adults. Or, excuse me, they rank only second to vitamins in popularity among adolescents. Some of the common ingredients that are found in energy drinks and energy shots, not surprisingly, are caffeine, not only from natural sources, but also synthetic caffeine. And some of those natural caffeine sources include guarana, yerba mate, and green tea. And some of the other more common phytochemicals that you'll find in energy drinks include synephrine, as well as yohimbine. What's interesting about many of the energy drinks is that the caffeine content is rarely indicated on the label. And not too surprisingly, caffeine content can vary considerably among the various brands of energy drinks. And of course, these are heavily marketed towards adolescents. There's no problem with that, but nevertheless, it's not uncommon to see events like the X Games that are sponsored primarily by Monster or some of the other energy drink brands. Nothing wrong with that, but nevertheless, they know the adolescents consume these products very heavily, and so they're marketed very heavily to them. And of course, it's much easier to drink many of these energy drinks rather than a hot cup of coffee or carbonated colas. And it's much more easy to slam down one of these energy drinks or energy shots as opposed to, again, a hot cup of coffee or a highly carbonated beverage like a Coca-Cola. And somewhat disturbingly is that many of these energy drinks and energy shots are very commonly mixed with alcohol. Now, as I mentioned, caffeine content is rarely indicated on the labels of energy drinks. And so what we're showing here is just the caffeine content for just a small sampling of the number of energy drinks and energy shots that are on the market. And so because the caffeine content is often not indicated, both consumers and healthcare professionals often really can't discern how much caffeine is being consumed. Some shots are highly concentrated and pose a greater risk for toxic ingestion. So, for example, I've highlighted some here in red, like 5-Hour Energy. There's 200 milligrams in just 2 ounces. The ALRI Hyper Shot, 500 milligrams in just 2 ounces. And one called Cocaine Energy Shot has 280 milligrams in just 2 ounces. So again, you can, by consuming more than one of these, you can start to get significant amounts of caffeine in a very short period of time. I think one of my most favorite is a product called Zombie Blood, which actually mimics, it's shown here on the left, it actually mimics an IV bag, and it has a biohazard sticker on it, so I thought that was pretty clever marketing ploy. Right now, as one might expect, side effects associated with energy drinks are primarily the result of the pharmacologic effects of caffeine. And so, as I mentioned, some of the very common side effects that you might experience after consuming an energy drink or an energy shot includes insomnia, nervousness, GI upset, nausea, tremors, elevated blood pressure, and such. But for the most part, these products are generally safe when they're consumed in moderation. It's when people start to go overboard that we start to see a lot more of the adverse side effects. And so the toxicity is likely a function of the dose or the volume of liquid consumed. Of course, it's also inherent upon the individual genetics of the consumer, as well as consuming these things in a very hot environment, or if an individual has a pre-existing medical condition, or if they're taking these products in conjunction with other prescription medications. It looks like the toxicity of energy drinks is increased in individuals that are relatively naive to caffeine, for also in individuals that are overweight and poorly conditioned with regards to their athletic capability. Energy drinks are also commonly mixed with alcohol, and this often leads to an increase in risky behaviors, and people are more likely to drive under the influence or binge drink or engage in risky sexual behavior when combining energy drinks or energy shots with alcohol. Now, energy drink use should be cautioned in adolescents that are taking medications to treat ADHD, or attention deficit hyperactivity disorder. And these include medications like Adderall or Ritalin. And so excess caffeine intake also increases the risk of heart-related illness because people have difficulty perceiving fatigue as well as hyperthermia when they're taking these things. And so that's why it's very important to be very careful when you're taking these things in a hot environment, because the stimulatory effects of the caffeine can often reduce your perception of becoming overheated or becoming fatigued. Now, as I mentioned, at one time, caffeinated alcoholic beverages were widely available, but they were actually removed from the market in 2011 by the FDA because of an increased risk of adverse events. Now, with regard to efficacy, it must be understood that energy drinks are not caffeinated Gatorade. Right? These products are not for rehydration or electrolyte replenishment, although many consumers treat them as such. Now, caffeine by itself does appear to have some ergogenic effects. Ergogenic effects means that it can improve athletic performance to some degree. And caffeine doses of 2 milligrams per kilogram may improve one's weightlifting ability or running or cycling times, but usually these effects are only noticed in trained athletes. The effect of caffeine on exercise performance, however, is not dose dependent. And so the more caffeine you take doesn't mean that you're going to be able to lift more or run faster. Usually there's an ideal dose of around 2 milligrams per kilogram that appears to be ideal. Many studies also show that caffeine can certainly improve mood and also increase feelings of alertness. So, in conclusion, multi-ingredient caffeine-containing products exhibit greater pharmacodynamic consequences than caffeine alone. Now, the pharmacology of complex formulations of naturally occurring stimulants is poorly understood and rarely researched. Activities like overindulgence, concomitant medications, underlying health conditions, environmental and genetic consequences can impact the safety of multi-ingredient caffeine-containing dietary supplements. And nevertheless, the efficacy of most multi-ingredient caffeine-containing dietary supplements still remains quite questionable. And so, as a result, with regards to the safety concerns combined with questionable efficacy, these types of products are oftentimes a recipe for disaster, especially in susceptible consumers. So with that, I thank you for your attention,